bom dia, good morning. Então, estamos retomando nossos seminários PAMB, né, e eu tenho aqui um convidado internacional. Eu vou apresentá-lo, aí já passo a palavra para ele. Okay, good morning, everyone. Today we have a pleasure to have Dr. Richard Bouchard here with us. I will briefly present him. So Richard, he hosted Diego during his time in the US. He supervised him in his research there, which was awesome for Diego and also for us to learn with him. Richard got his bachelor degree in biomedical and electrical engineering. If I'm wrong, Richard, you can stop me and correct me, please.
Sounds good.
I saw that you also got a bachelor in cultural anthropology.
Yes.
All of them from Duke University. And then you got your PhD. He got his PhD also from Duke University in biomedical engineering. And then he completed a postdoctoral fellowship in the ultrasound imaging and therapeutics research laboratory at the University of Texas at Austin. Then he moved to the MD Anderson Cancer Center, University of Texas, where he's now an associate professor. His research interests include preclinical and clinical photoacoustic, ultrasonic imaging, and ultrasound-based elasticity imaging. It's really very nice you are here to present your work to us, Richard. Now, the stage is yours. Thanks again.
Great. Can you hear me all right?
Yes.
Okay, cool.
Perfect.
Great. Thank you for the kind introduction. And yeah, Diego's actually got, there'll be a couple papers that I'll show work from that he's actually got authorship on, including one we just got accepted. So Diego, if you're listening, that was a couple days ago. So pretty exciting. Okay, so I'll jump right in. So start with just some disclosures. In the US, we're all about disclosures because you don't want to get sued. So no financial interests or relationships to disclose. And this really is all preclinical, so not FDA approved. Just a little pitch for MD Anderson Cancer Center. You can see this is actually, we're part of Texas Medical Center, which is 5.4, I had to look it up in kilometers. It was in square miles, so kilometers squared. Over 106,000 people work in the medical center, so it's pretty massive. MD Anderson's the largest institution in the medical center. Just to give you an idea, this is MD Anderson building. It's like 20 stories tall. Looks pretty cool. This is just for administration. So this is not, they don't see any patients, nor is there any research done in this building. It's just a bunch of, as we call them, bean counters. I am in Department of Imaging Physics, but as Teo said at the beginning, I'm an engineer. My lab focuses on ultrasound-mediated imaging. I have a couple different roles at MD Anderson, and so I work with a variety of investigators. If anyone has an opportunity to come to Houston to work in the medical center, I'd strongly recommend it. But we're also home, perhaps more famously, to NASA. So that's pretty cool. You can see the Saturn V rocket. So anyway, that's my little pitch. It's not quite as beautiful as it looks like based on the intro video that was played as your campus, but we do have some cool stuff in the city. One last little pitch. This is for our small animal facility. So we have perhaps the largest one in the world for imaging. So my chair is the director of the facility. We get NIH funding. We have capacity for about a third of a million mice. So it's quite large, and we offer every modality. We've got 4 or 5 MR systems, multiple PET, multiple CT systems, ultrasound, and then tons of optical systems, as well as other modalities, radiation therapy, all for mice. So it's pretty impressive. Okay, so now I'm going to jump right in. So I did my graduate work in biomedical engineering. I'm really an ultrasound person. And so I think the question, just to kind of start, and you'll see this talk, I'll kind of focus in, then focus out a bit, and then focus back in. But the question is, why ultrasound? So we're often kind of trying to jockey for position with other modalities. And so here's a couple things that a lot of people don't realize about ultrasound is it has incredible temporal resolution. So we're talking 20 kilohertz full frame, which is possible on this commercial system to the right. It has very impressive spatial resolution at depth. And I'll say that in quotes because at that resolution, you're probably going to be limited to, maybe 10 or so millimeters. But depending on your background, if you have an optical background, 10 millimeters is like 100 kilometers. So impressive spatial resolution, much less or less than 100 microns. In certain cases, significantly less than that. Good penetration depth, greater than 10 centimeters. And put a little caveat here that I realized you can't concurrently achieve all of these. These are sort of best operating case, but they result in trade-offs. And the physicists in the audience don't really have to explain that. So you can't, if you trying to achieve optimal resolution, then you'll get inherently more attenuation. So that'll hurt your depth penetration. But anyway, these are kind of all best case scenarios. This one I particularly like. So as you've probably seen in the movies, the US loves their military. And so this is actually in the field, an ultrasound system. And so not only do we not require a Faraday cage and you name it, and, or shielding from radiation, we can actually achieve ultrasound imaging just about anywhere, even in the battlefield. And so it's really quite easy to use, portable, non-ionizing. That is the shielding for the Naval Research Laboratory cyclotron. It looks like it's, a couple meters thick. And so pretty crazy, low cost. So this is from eBay, which I'm guessing exists in Brazil or some similar equivalent. And this is not something you'd use on a human, but just to give you an idea, this is actually for horses, and you can buy this system for $300. So pretty cheap. You can buy clinical systems now, though, in the thousands for really particular indications, but, a couple thousand. Meanwhile, a textbook for CT MR will cost on the same order. So very low cost. Okay, well, just to be honest and sort of balanced, why not ultrasound? Well, ultrasound can't do everything, although I'd like to convince my students it can. It only has adequate soft tissue contrast. In some cases, it becomes quite difficult actually to delineate certain pathologies compared to, let's say, MR, which has quite good soft tissue contrast. Anatomical with limited functional. So really the most ubiquitous functional would be Doppler. This is sort of for standard ultrasound, if you will. Restricted to intravascular contrast or microbubbles. And it's not particularly quantitative. So there are quantitative modes that are being developed, but it fundamentally is a bit difficult. Essentially, if you're relying off of the amplitude, which is what brightness mode ultrasound essentially is, there are a lot of things that can confound that and can make it difficult to quantify. It also can't image through bone or air. There are folks that are working on this and have achieved certain applications that sort of can skirt this limitation, but generally it's something that is kind of a deal breaker. But these limitations can be addressed in part by what is termed, and I like to use this term, ultrasound-mediated imaging. So I'm going to come back to this little animation a couple times. So essentially, we've got a region of interest that we want to interrogate. So normally with pulse echo ultrasound, you're going to transmit a pulse of sound, you're going to get the backscatter, the echo back, and you're going to make an image of that, which you can see here on the right. With ultrasound mediated, we will often also do that. So we can, in a lot of cases, still implement traditional pulse echo ultrasound. But the question is, what if we induce a local mechanical perturbation to the region of interest and then apply conventional ultrasound imaging methods? So for elasticity imaging, we could induce gross motion. We could also induce cell molecular specific acoustic source, or create them sources or scatters. And this is what we do with photoacoustic contrast, or when we activate nanodroplets. So I'm going to go into kind of all of this later in my talk. So advantages, ultrasound mediated, it often can use the same or similar hardware and even algorithms that have been developed for conventional B-mode imaging. You can get increased contrast. This is work that we did a long time, about 10 years ago now. This is a brachytherapy seed, so you can barely see it on the left. In the ultrasound image, you get really nice contrast. And then you can actually see, kind of appreciate the marriage of the two. So photoacoustic ultrasonic imaging, whereby you have high contrast of certain optically absorbing entities, but overlaid B-mode for soft tissue. Quantitative tissue characterization, so this is SO2 in the femur of a mouse, and molecular imaging with nanoscale agents. So this is lipo-JACG, which I'm going to show in a little bit. There's a number of different ultrasound-mediated techniques, probably even more now. I'm really going to focus on two of them. I did all of my graduate work, actually even started when I was an undergrad, working on ARFI-based shear wave imaging, elasticity imaging, but I'm not going to show any of that work. Okay, so now that I've kind of given my pitch for ultrasound, I'm going to take a step back and kind of give you a little bit of context, clinical problem. So what's the problem? What are we really trying to address with molecular imaging? And there are a number of different examples. And in fact, I'm doing a bit of work, and I've done, in the past, I did all cardiac work. Since I've come to MD Anderson, Anderson as an institution isn't as interested in maybe cardiotoxicity. There are folks working on that, but essentially if it doesn't relate to cancer, they're not particularly interested. But now I've started doing some work in cardiac domain with Texas Heart. And so a lot of this stuff that I'm going to talk about can be applied to other pathologies, but my focus today will be on cancer. And so this kind of shows, this is a CT that gives you an indication sort of ultimately when we're trying to find tumors early, often they're just far too small to be detectable with conventional imaging methods. In addition, the actual manifestation and the growth of the tumor is sort of downstream of the initial sort of genesis, if you will, of the cancer itself. So it's pretty kind of late stage by definition. In addition, if you can see the tumor and you're trying to use it, for instance, for therapy monitoring, it's not necessarily always correlated. The change in tumor size not always correlated with treatment response well, and it too is often, it by definition is sort of downstream. So if you're waiting to see a change in size and it doesn't happen, it might be too late to change course on therapy. real briefly, I'm not going to really go into this, but essentially there's a lot going on in the microenvironment of a tumor. And a lot of this is still not terribly well understood, but there are environmental conditions that have established certain biomarkers or emerging biomarkers of what's called the tumor microenvironment. And so this gives an opportunity to establish a imaging biomarker to monitor tumor growth. Right now, we tend to use histopathology. This is really the standard. This works quite well in a number of cases, and assuming you can harvest the proper tissue, then it's pretty good at characterizing that tissue. But harvesting the tissue can be a significant problem. Often it's done somewhat randomly with biopsy. The biopsy doesn't necessarily harvest the tissue that one might intend to harvest, and it doesn't really easily permit longitudinal sampling because you have to go in and take a biopsy, which can also have its own complications as well. So the question is there another way? So the sort of traditional paradigm with biopsy, it's gotten us a long way, and I don't think it's going anywhere anytime soon. But is there another way that we could, with imaging, achieve something that's non-invasive, that's high resolution, quantitative, volumetric, longitudinal, sensitive, and potentially in real time? And this, I believe, can be achieved with molecular imaging, which could bridge traditional in vivo imaging with ex vivo assays. So current molecular imaging, there's some preclinical modalities, and those have actually been developed perhaps the most. And those tend to use optical methods, some that require ballistic photons, so those have depth limitations, that you can make windows and such, and others that don't, but really don't provide good resolution. One can almost say doesn't really provide resolution at all. It's kind of a blob. Clinical molecular imaging techniques, the most ubiquitous one would be really PET-based, which is ubiquitous at MD Anderson, works quite well, but it's incredibly expensive, very difficult to implement. We're lucky we actually have a cyclotron in this building, but making radionuclides is difficult and requires very specific equipment, and it's ionizing. So it's really not something that you have to be considerate when you're thinking about implementing this. fMRI, there's DCE bold. Some of these work better than others. Hyperpolarized MRI, which is being developed at Anderson as well as some other institutions around the world. And these can achieve some of what we want, and I sort of bolded those there, but it really doesn't get every single bucket. And so the question, is there another way? And voila, photoacoustic imaging, which I'm going to explain in one moment. So it's non-invasive, high-resolution, quantitative, volumetric, longitudinal, sensitive, and it can be achieved in real time. And so some people joining in may wonder, well, what the heck is photoacoustic imaging? Well, it started with really Alexander Graham Bell and Charles Tanter, but no one remembers him. Alexander Graham Bell is quite famous, particularly in the United States, has other inventions. He's the one who discovered this. Essentially, you can think of it as analogous to lightning and thunder. So you irradiate, you flood the tissue with photons. They not only do they not need to be ballistic, it actually works better if they're not, if you get scattering, so it spreads the photons out. Those photons then interact with absorbers in the tissue, hemoglobin, for instance. This light is of very short duration, and so it will absorb, it will thermoelastically expand, and then it will contract because it is short duration pulse. And so when you have this expansion contraction, that is a local acoustic source. So you have lightning, and then you have thunder. So back to our little example, PAUS imaging. In this case, we're going to irradiate. It really doesn't even matter the direction of the irradiation. Sort of, there's actually caveats to that, but because you're not relying on ballistic photons, the direction doesn't really matter as one might expect. And so we can then get an image that's proportional to the optical absorption of the absorbers in tissue, and we can add that to beam mode ultrasound. So we can get one on top of the other. And so this is showing sort of an example of this. I apologize, the slide's a little messed up. Yeah, and we can get, based on optical contrast of absorbers, so hemoglobin, melanin, water, kind of, although that has a huge caveat, as well as exogenous agents, nanoparticles, absorbing dyes. What makes this really powerful is we can then implement multi-wavelength PA imaging. And so this is something that's really standard in optical-based really imaging techniques, is that you can then image at different colors of light, typically in the low near infrared, and get the response at those different colors. So you can get what I would call a PA signal spectrum, and you can compare that to known optical absorption spectrum, and you can implement different methods to deconvolve that into different species of absorbers that are assumed to be in the field. You can do linear unmixing, for instance, is the sort of most straightforward implementation. Now, what do we need for clinical PA imaging? There's actually some debate on this, particularly as to how deep. This is a question I always get. So if anyone asks me this at the end, my answer will be somewhat qualified. No one really has a great answer as to how deep photoacoustic imaging can image. It can definitely go a few centimeters if properly done, but even that can be difficult. And so we're really focused on somewhat superficial organs such as the breast, or organs that we can reach endoscopically, such as the prostate and such. There are a few clinical trials in the United States going on. At the time when we got these data, it was 37. Most rely on blood. And so only 5 were relying on an exogenous agent, and that agent was non-targeted IR dye 800. And so we need to be able to image greater than a centimeter, and we'll make a case in a little bit that we probably need to have exogenous agents, or availability of that, particularly if we want to achieve quantification. Now, these are the systems we have at MD Anderson. We've got a Visual Sonics Vivo Laser 2100. We got this on an NIH S10 grant. We have an iThera Medical MSOT, which is a photoacoustic only. We also got this on an NIH S10 grant. This is in my lab. It's a quote unquote clinical, and actually we've got a couple new transducers that aren't shown here, and another laser source. And this is usually the image it makes, the blue screen of death. It has a lot of flexibility, but it's not always the most stable. And then lastly, and this is really owned by Marty Pagel, a collaborator at MD Anderson. So we have a iThera Medical Acuity. So this is a clinical, in quotes, PAUS system, but it's not yet FDA approved. Okay, so just to reiterate sort of why bother, photoacoustic imaging can kind of bridge the gap. So it can achieve, and it can really, ultrasound in theory could actually go lower. It typically doesn't, but it can go a bit lower than this. But the way that we can kind of get this really exquisite resolution is through the use of contrast agents that were typically used for optical imaging. So we can ostensibly achieve really good resolution at pretty good penetration depth. So it's sort of a balance of optical imaging and ultrasound. And I've kind of mentioned a bunch of this. And excitingly, it recently was FDA approved, actually at the beginning of this year. This was in part done with a study at MD Anderson. They were one of the main sites. I was tangentially involved in it, and it's through Seno Medical, and it's for breast, label-free breast. So that's pretty exciting. Okay, now I'm going to this is sort of going to frame specifically the foci that I have in my lab and what I think is really necessary for photoacoustic imaging. Photoacoustic imaging has a lot of potential, as you can see, and it's already made its way into the clinic with one approved application. But there's still some issues that need to be addressed. And so this meta-analysis is a little bit old. I did this in mid-2019, but I suspect the results still hold true. So if you look at, this is from Web of Science, you can see I don't necessarily know if I call it exponential growth, explosion of publications, but a real significant monotonic increase in publications, which is exciting. Now, if we look at those with just that used exogenous agents, so this would be total that use photoacoustic imaging for biomedical applications. This is those that use exogenous agents, and these are those that used organic contrast. What we see, if we zoom in on this, is that most don't use exogenous contrast. So most use endogenous. They looked at blood, is what happens 99% of the time. Again, there's some melanin work that's been done, but most of the time it's hemoglobin. What we see, so a minority of the publications are exogenous contrast, and then a minority of those are organic contrast. And then if we focus in on 2 years, 2009, in 2009, 65% of the publications were under a centimeter imaging depth, 35% over a centimeter. And going back to an earlier slide, we really need to be over a centimeter, more than likely to be able to hit a lot of these clinical applications. In addition, and I'll make this case in a little bit, I think we're really going to need exogenous agents. The problem, though, is most of the work right now is being done on inorganic agents. Inorganic agents typically have a much more difficult regulatory burden. They're really cool in nature, but they're not necessarily the most clinically relevant. And then if we hop over to 2019, 10 years later, we've actually decreased in imaging depth. This is, I believe, due in large part to the commercial availability now of small animal systems, which really focus on murine models, which tend to be pretty shallow. And so some of the key points is that most imaging research focuses on blood, endogenous. So this is important, we believe. It allows for label-free imaging, cheap and easy, could provide insight into extracellular hypoxia. And then I'm going to hit these other points in a moment. So I'm going to jump into sort of 3 vignettes quickly to give you kind of an idea of some of the work we've been doing. So this was done a little while ago, published in 2019. Diego is actually on this paper, so I'm going to point out every publication he got on. So I think he's maybe 3rd or 4th author on this. And this was work done with Marina Konoplava's group. And I'm going to go through it pretty quickly. Feel free to ask questions on it because I've still got, as you can tell, a number of slides, and I don't want to run over on time. So this was for the purpose of using photoacoustic imaging to try and originally assess hypoxia in a leukemia model. When I first started in photoacoustic imaging, I didn't really understand what hypoxia was, and was a misnomer really in the field, and there still is in some circles, that we can image hypoxia. Well, we don't. so hypoxia would be is typically referencing extravascular. It could be intracellular or extracellular, but it's typically extravascular hypoxia. And it's the partial pressure of oxygen. So we're not directly sensitive to that at all with label-free photoacoustic imaging. What we're sensitive to is SO2. And so that's going to be directly related to the intervascular hypoxia, which is, again, not exactly what we're interested in, but we're able to then characterize the percentage of hemoglobin bound to oxygen. And so hypoxia, extravascular hypoxia, is correlated with SO2, but it can get a little bit complicated sort of the further you get away from the vascular source, because there's a lot of unknowns that can confound this correlation. And so as time went on, myself and others really focused more on just developing SO2 as a biomarker itself. And so all of the work I do now is murine, they're murine models. So this was no different. Murine model, this is the workflow of this particular study. Again, for the sake of time, not really going to get into the nitty-gritty details, especially of this study that was done a little while ago. We often use other modalities. In this case, we used optical imaging to discern tumor engraftment. We then imaged photoacoustically. We also imaged with CT, small animal CT, and we co-registered so we could determine the ROI that was in the femoral bone marrow. So you can see it right here. So we're focused really on this region right there. And so that's what this is primarily done by Kayla Wood, former grad student who's now doing her fellowship in medical physics. And we did repeated measures to look at SO2 and got pretty low variability, although there was a dependency along the femur as to the absolute SO2. We then, so this was through time. And through time, what we saw is that we saw a change in the control mice versus the leukemic mice in photoacoustic-based SO2. And so this is essentially the difference, the absolute difference. And there's sort of a couple different hypotheses that we had to explain why we think things were changing. It's not always intuitive, though, to kind of discern changes in SO2, because there's a lot of different things, particularly downstream. So if things change downstream where the oxygen's normally being consumed, or if it's being consumed more vociferously, then it will affect what you see at the source. And so this was some work basically kind of showing that SO2 has potential to differentiate between different pathologies, in this case, leukemia. So key points revisited. So most PA imaging researchers focuses on inorganic contrast. So virtually none of these agents is in clinical trials. Organic contrast, in terms of the organic contrast would have significantly reduced regulatory concerns. So this work is done with Konstantin Sokolov, who's my main collaborator, and Anil Sood. We've actually since gotten a CPRIT on this and an R01 on all of this work. And excitingly, we got, and Diego is actually on this. He doesn't even know this was accepted, but we got accepted about 4 days ago into Nature Communications on this. So it should be available. We're actually sending the proofs back tomorrow. So it should be available on Nature's website, hopefully end of this week. But this technology is essentially liposomes that we put a high concentration, or rather ICG and high concentration within liposomes, such that they J aggregate stack, so they stack on top of each other, which gives them really unique optical properties. I'll show you in a second. And then we can conjugate the liposome for targeting. And so this is spectrum of blood. Typically, ICG is here, which is really kind of annoying because it's right where blood changes or hemoglobin changes quite a bit. Not to mention that ICG is actually the spectrum is concentration dependent, so this can shift. Lipo-GACG, though, is stable because it's encapsulated in a liposome, and we have a red shift. So hemoglobin stays pretty constant. So we really have improved unmixing accuracy between lipo-GACG and hemoglobin and reduced regulatory concerns. All of the constitutive components are FDA approved. The construct is not, but all the components are. It's nano-sized, it's stable. Again, if you're interested in this, the preprint should be available really soon. There's a whole ton that I'm not going to show you. We had to do quite a bit for this paper in terms of characterization and toxicity, but it's linear, it's stable. This was actually some of the work that Diego did. So we characterized in tissue-mimicking phantom with different titrations of blood and then concentrations of ICG or lipo-ICG. So we could actually quantitatively assess sort of the accuracy improvement. We also implemented fluence correction, which I'm not going to go into, just that this is the third paper that Diego actually got on. I think he was maybe second author on this. And so this is the comparison in phantom. What we see, essentially what this is looking at is if we have different concentrations of our probe, either ICG or lipo-ICG, how well are we able to discern the SO2 in that region? And so what we see is that if we have high concentration of our probe, we get really significant SO2 estimation accuracy with ICG. But with lipo-ICG, it stays pretty low, under about 2%. So this is good because we want to be able to do both. We want to characterize SO2, but also characterize our probe concentration. And then this is looking at if we increase our probe concentration in different SO2 of blood, how stable is that? And what we see is that for lipo-ICG, we actually get a really good ratio, whereas that's pretty accurate, pretty similar to the concentration ratio in terms of the unmixed probe signal. Yet for ICG, it can be drastically off. In this case, we have no ICG, yet it thinks that we've got half of what we have at 0.5 OD. So we're a lot more accurate. This is toxicity. This is in vitro, so targeting folate receptor alpha in SCOV3 cells. For the sake of time, I'm going to kind of go through this quickly. We then showed in an in vivo model, SCOV3, orthotopic. So this was at the site of the ovary. We did this in conjunction with MR. So this is MR right here. You can see the tumor. This is sort of depicting the full workflow. We did multi-wavelength volumetric imaging. We then unmixed for both lipo-GACG and SO2 at different time points pre and post injection. We then looked at the contrast enhancement pre and post injection. And what we see is that the SO2 in all of the organs in the tumor we looked at does not significantly change pre or post injection. This was, I thought, the most exciting result because oftentimes if you introduce a contrast agent of high enough concentration that you're sensitive to it, the downside is that you will confound your SO2 because it's sort of, you get crosstalk in the spectrum. But this suggests otherwise, that we could get independent and really fairly sensible measurements. And what the editor liked the most is really this. This showed that we get accumulation in the liver and spleen. We didn't see any in the kidney, which had actually been consistent with some other work done with liposomes. And then in the tumor, we get enhancement in both targeted and non-targeted. We had one targeted with folate receptor alpha, one that was conjugated to RG16, so it's non-specific antibody. And what we saw as time increased is you saw the targeted became significantly greater accumulation than the non-targeted. We also demonstrated this sort of at clinical depth. We essentially made a mouse hamburger, if you will. So we had local injection of the agent, but then we put a standoff that was 1 centimeter, and then we imaged our agent. So this is lipo-JCG. You get really good signal. ICG, not very good. We imaged it with this clinical MSOT system just to kind of show the potential for future clinical application. Okay, so, and I'm going to whiz through these so we have time for questions. So this last point is that most PA imaging research, as I showed at the very beginning, focuses on non-clinical depths. So in 2019, 85% was less than 1 centimeter. So perhaps there's another angle we can take a look at. And really that limitation is fundamentally because you're using light. And even if it doesn't need to be ballistic, your fluence will precipitously drop off with depth. And so maybe we can look at an ultrasound-only molecular imaging solution. One of the real assets of this type of a platform is that it can be implemented potentially with technology already in the clinic. This work is done also with Konstantin Sokolov as well as Stephen Lai, who's our clinical collaborator. So in this case, we're going to have a nanodroplet, which is really a kind of a condensed microbubble. It's nano size. We're then going to insonify with ultrasound. So we're going to expose it to a sufficient negative pressure such that it will convert to a microbubble. We can then obtain a nonlinear ultrasound image. So we'll essentially have a microbubble, which is now used pretty commonly in the clinic, but we've have potential to deliver it extravascularly. So we can introduce it as a nano-sized agent. It can then extravasate out of the vasculature, and then we can activate it and convert it into a microbubble. So this really gives us potential to kind of skirt the diffraction limit of ultrasound. You can't fundamentally really image nano-sized things because of the wavelength of ultrasound, but if you can just introduce them as nano-sized and then convert them into micron size, you can sort of skirt that. I'm going to kind of jump through some of this. And so this is an example of what some of the work, and this is actually all Trevor Mitchum, who is currently looking for a job. He's actually got a couple interviews in the next few weeks, but a recent grad from my lab, so Dr. Trevor Mitchum. And so this shows you sort of how it works. We started about nominally 200, 250 nanometers. Upon ultrasound activation, we then get to larger than a micron. And so we activate at different locations in our field of view. And I wish he made this bigger, but you can see the region along the point spread function that we actually activate. This is doped with nanoparticles or nanodroplets. And so we only get contrast where we activate and have nanodroplets. We don't have nanodroplets in the standoff, for instance, overhead. These can be targeted. He actually just sent me the draft yesterday. We're about to submit this manuscript. So we've done a bunch of characterization. We've also conjugated them, looked at different mixtures. We tend to like C6 particles. This is our lab system. We've got a fairly high resolution CMUT transducer with Verisonics platform. For the sake of time, I'm not going to really go into this, but we get really nice signal with this particular C6 formulation that we've developed. This is a Phantom experiment. So because we're University of Texas, if you can see it, it's UT. And so here we UT. We also show this in vitro targeting. These are EGFR targeting FADU cells. And so pre-activation, we get nothing. Post-activation, we get, with unlabeled cells, we get nothing. With pegylated droplets, so essentially non-targeted, we get much less. And with targeted, we get significantly more. This is showing in some of our early in vivo model. And so we inject here and here, and looks like he did not. What we first activate right here, and then we activate right here. This is showing after both activation, but you can see them light up separately, which is pretty cool. So we can, this is from local delivery, but we can achieve that in vivo. We've also now included size 7, so we can optically track. So this is our nanodroplet actually in the tongue, kind of a crappy image, but in the tongue. So this is a photoacoustic image. We can also then look at it ex vivo. We've got, and I think we're the first site maybe in the world or US that have one of these Zeras. So it's a specimen. It will slice and do a white light and then fluorescence of every single slice in a specimen. So mouse, make data sets upwards of, it's like 0.5 terabyte or something insane. If you do the full volume, full resolution, all the channels, everything. But it's really, really cool. And because size 7 is really both a fluorophore and a chromophore, we can implement fluorescence imaging to kind of figure out where it goes. Not really going to dwell on that. Ultimately, we think we can use the Vivo MD, which is Visual Sonics' fairly new high-frequency system to implement this. So conclusions, we kind of jump through this. Photoacoustic-based SO2 imaging, I think, shows a lot of promise. There are a number of groups working on this, and it provides something that's related to SO2, not hypoxia directly, but label-free. Photoacoustic imaging targeted lipo-JCGs, we're really excited about this, and we've got multiple applications. We've also got a study now on neoadjuvant monitoring of neoadjuvant chemotherapy for triple-negative breast cancer. And then nonlinear imaging of activatable nanodroplets. Again, there are a number of groups working on this now too. I think it's most exciting because it can use sort of already available hardware. So acknowledgements, a number of people. I wish I had Diego. So Diego, I absolutely can't forget him. He was involved in bits and pieces of a lot of this, as well as Kosha, Dr. Sokolov's lab, and some other labs. Our funding sources, particularly NIH, I would like to thank. And that's it. Fini.
Hi. Okay. So thanks for the very nice talk, Richard. So I had the opportunity, I didn't say that, but I had the opportunity to visit your lab, and the structure you guys have there is really awesome. And hopefully next time you're going to be able to come here and talk in person.
I would love that. I would love that.
This style is very nice. That now during the pandemic, lots of people are doing this type of interaction, which is good, but in person I think is much better. So now we are open for questions. If you want to, se você quiser fazer a pergunta em português, fique à vontade, eu posso traduzir. So if you want to make your questions in English, in Portuguese, I can translate that, but otherwise you can just go on and write in English.
And if Diego's if he's watching, he's got to ask me at least one question.
Yeah, Diego, please, you have to make a question.
And it's got to be in English.
Okay, so I think we got one here. So Luis Henrique Nunes, he said that a great presentation. So when you excite the nanodroplets, if they are charged, could there be some interference on the signal?
Electrically charged, I'm guessing, is what you're referring to. So yeah, we haven't really looked at that. I mean, other than maybe for the cell interaction with zeta potential and such, I guess that could matter. That's not in preclinical. In the phantom stuff, we really haven't looked at the influence of a charge on the particle itself. But you bring up a great question just on the interaction of the droplets, and that's something we're looking at in terms of coalescing, for instance, which is a problem because that would then change the size of the droplets. And so that's something you wouldn't want. But we haven't really kind of figured out all those details. The dynamics can be pretty complicated at that small scale.
Thanks. So I think we get another one here. Guilherme, thanks for the call.
I'll try and be brief. So
could you please explain about the fluence correction?
Yeah. Originally we were implementing a rather complicated fluence correction method whereby we essentially would, with finite element modeling, we would kind of make certain assumptions about our field and constrain the scattering that really had to be fairly uniform in our field, but then allow iteratively the absorption to update until we essentially got a convergence of this model-based approach. That one is incredibly computationally expensive, and in the end, I don't, because you don't still know much of anything about the scattering, I don't really know what it bought us. And so now we've kind of taken a step back and actually implement fairly simple fluence correction whereby we're just trying to get the surface fluence, so through the water, for instance, which is well known. You can make assumptions about the standoff, for instance, and then really not making too many assumptions about the underlying tissue other than kind of assuming a mu effective. So just a estimated mu A and reduced scattering.
Are you still using the finite element? Guilherme is now working with like Monte Carlo's ways to do something similar. That's
we still do, only because at this point we're not like too fixated on the time it takes. But yeah, you bring up a good point. I mean, it's probably, and actually what we actually need it for now, we could probably do something a little bit more straightforward.
I guess Diego is here. He's making questions. He has a big mask there.
I have yeah. Hey, boss.
Is there any study that follows radiotherapy treatment in vivo and how?
Yeah, there's been a couple.
With the skin.
Yeah, so especially because of the correlation with hypoxia and sensitivity to SO2 and dependency of hypoxia on sort of efficacy of radiation therapy. There definitely been studies. I haven't been particularly, I just don't have the bandwidth really to do it. And melanin and skin. So this is an interesting question, and one that is a bit regional, particularly with, I would say, sites in the Americas, North America and South America, versus some places in Europe where some places in Europe, they don't have as many people in their population that have sort of the darker pigment in their skin. And that definitely affects a whole slew of, it can cause spectral coloring, and just on a first order, it just, reduces your signal, so your penetration. So there's no real magic trick to it. If you have darker skin, it's generally not going to be as ideal.
Okay, nice. Probably we can have final questions. Professor Adilton here is asking something about breast photoacoustic tomography.
Yes.
Have you ever worked with photoacoustic tomography?
So not what you're probably, so for instance, the MSOT is technically it's tomographic because it's an arc array. But some of the really cool, so Kruger had some of the awesome breast images that sort of showed angiography. And then Paul Beard has sort of more recently, with his very unique system, made some absolutely exquisite, not quite as deep as what Kruger showed. So no, I have not sort of achieved those super high quality, but those are fairly niche, super, super cool. Unfortunately, my systems tend not to make quite as pretty a picture, but it's sort of a trade-off with imaging depth and what you can interrogate, and multi-wavelength.
Okay. Just one question from me related to the leukemia model. So what was like the rationale? Is that a common thing that leukemia bones that they're going to become more with hypoxia? Although as you said, you're not measuring actually hypoxia.
So that was actually demonstrated by Quantitative with intervital imaging is what they used, and they showed there's hypoxic niches that develop in the femoral bone marrow. And so, and they were actually, and Quantitative, I believe this is, it's either still in clinical trial or it went through a clinical trial, a therapy that targeted hypoxia for leukemia. And so that was the sort of motivation, and they wanted something that was label-free and non-invasive. So the idea was, can we look at SO2 instead, which would be non-invasive and label-free.
So, and in your opinion, in this case, can we make the assumption that the SO2 was well correlated with hypoxia?
Yeah, I mean, well, SO2 is going to be almost perfectly correlated with intervascular hypoxia, so partial pressure of oxygen in the vessel. As you start getting further away from the vessel, though, other things, it becomes more complicated.
Okay. Okay, Richard, I think we don't have further questions here in the chat. It was very nice. So I think now we can close the session.
Awesome.
We can talk a little bit more. We are going to close here the transmission, but we can like finalize here afterwards. Just me, you, and Vivian. Okay, so obrigado todo mundo, and have a good end of week. Okay.
Yeah, thank you, everyone.